welcome to Biology, the scientific study of life. More about the cell. The eukaryotic cell's genetic instructions are housed in the nucleus and carried out by the ribosomes. When the first stop of our detailed tour of the cell, let's look at two organelles involved in the genetic control of the cell. The nucleus, which houses most of the cell's DNA, and the ribosomes, which use information from the DNA to make proteins. Nucleus genetic library of the cell. The nucleus contains most of the genes in the eukaryotic cell. Some genes are located in mitochondria and chloroplasts. It is generally the most conspicuous organelle in the eukaryotic cell, averaging about 5 micrometer in diameter. The nuclear envelope encloses the nucleus, separating its contents from the cytoplasm. The nuclear envelope is a double membrane. The two membranes, each a lipid bilayer with associated proteins, are separated by a space of 20 to 40 nanometer. The envelope is perforated by the rate pores that are about 100 nanometer in diameter. At the lip of each pore, the inner and outer membranes of the nuclear envelope are continuous and intricate protein structure called a pore complex lines each pore and regulates the entry and exit of certain large macromolecules and particles. Except at the pores, the nuclear side of the envelope is lined by the nuclear lamina, a net-like array of protein filaments that maintains the shape of the nucleus by mechanically supporting the nuclear envelope. There is also much evidence for a nuclear matrix, a framework of fibers extending through the nuclear interior. Within the nucleus, the DNA is organized into discrete units called chromosomes, structures that carry out the genetic information. Each chromosome is made up of material called chromatin, a complex of proteins and DNA. Stained chromatin usually appears through both light microscopes and electron microscopes as a diffuse mass. As a cell prepares to divide, however, the thin chromatin fibers coil up, which means condense, becoming thick enough to be distinguished as the familiar separate structures we know as chromosome. Each eukaryotic species has a characteristic number of chromosomes. A typical human cell, for example, has 46 chromosomes in its nucleus. The exceptions are the sex cells, eggs, and sperm, which have only 23 chromosomes in humans. A fruit fly cell has 8 chromosomes in most cells, with 4 in the sex cells. A prominent structure within the non-dividing nucleus is the nucleus plurinuclearly, which appears through the electron microscopes as a mass of densely stained granules and fibers adjoining part of the chromatin. Here, a special type of RNA called ribosomal RNA, small RNA. Notice that RNA small, this one means ribosomal RNA. M small, mRNA, which means messenger RNA. RNA or ribosomal RNA is synthesized from instructions in the DNA. Also, proteins imported from the cytoplasm are assembled with RNA into ribosomal RNA into large and small ribosomal subunits in the nucleus. These subunits then exist in the nucleus through the nuclear pores in the cytoplasm. There, a large and small subunit can appear to the cytoplasm, where a large and small subunit can assemble into a ribosome. Sometimes we are 2 or more nuclearly. The number depends on the species and the stage in the cell's reproductive cycle. Recent studies have suggested that the nucleus may perform additional functions as well. As we see in this picture, the nucleus directs protein synthesis by a synthesizing messenger RNA, mRNA, which means according to instructions provided by the DNA. The mRNA is transported to the cytoplasm via the nuclear spores. Once an mRNA molecule reaches the cytoplasm, the mRNA is then transported to the cytoplasm via the nuclear pores. Once an mRNA molecule reaches the cytoplasm, ribosomes translate the mRNA's genetic message into the primary structure of the specific polypeptide. Ribosomes, protein factors in the cell. Ribosomes, particles made of ribosomal RNA and protein, are the organs that carry out protein synthesis. Cells that have high rates of protein synthesis have a particular large number of ribosomes. For example, a human pancreas cell has a few million ribosomes. Not surprisingly, cells active in protein synthesis also have prominent nuclei. Keep in mind that both nuclei and ribosome, unlike most other organelles, are not enclosed in membrane. Ribosomes build proteins into cytoplasmic locale. Notice this one. Notice the picture. Notice. Okay. Free ribosomes are suspended in the cytoplasm in the cytosol, while bound ribosomes are attached to the outside of the endoplasmic reticulum of nuclear envelope. Most of the proteins made on free ribosomes function within the cytosol. Examples are enzymes that catalyze the first steps of sugar breakdown. Bound ribosomes generally make proteins that are destined either for insertion into membranes, for packaging within certain organisms such as lysosomes. Notice that in this picture. For export from the cell secretion. Cells that specialize in protein secretion, for instance, the cells of the pancreas that secrete digestive enzymes, frequently have a high proportion of bound ribosomes. Bound and free ribosomes are structurally identical and can alternate between the two rules. The cell adjusts and the relative numbers of each as metabolic change alter the types of proteins that must be synthesized. Okay, let's talk about this picture, the nucleus and its envelope. Within the nucleus are the chromosomes, which appears as a mass of a chromatin DNA and associated proteins, and one or more singular nucleus, which function in ribosome synthesis. The nuclear envelope, which consists of two membranes separated by a narrow space, is perforated with pores. Notice that these pores. And lined by the nuclear lamina. Close up. Notice the Bohr complex. Closer look. Close up of nuclear envelope. Rough ER. Notice this one, blue. The surface of nuclear envelope, TEM, of a specimen prepared by a special technique known as freeze fracture. Closer look. Notice that Bohr. This is the surface. Notice Bohr complexes TM. Each Bohr is ranged by protein particles. This one, inner membrane, outer membrane, nuclear Bohr, and this chromatin. Notice that chromatin, a nucleus, also a nucleus. Notice in the middle. and this pore complex. Closer look, this one, a close-up of nuclear envelope. Boom. This pore, nuclear pore here. Okay. Also, closer look of ribosome, diagram of ribosome. Notice that. Large subunit and small subunit. Notice ribosome, this electron micrograph of part of a pancreas, so shows many ribosomes, both free in the cytosol and bound of the endoplasmic reticulum. The simplified diagram of a ribosome shows its two subunits here. Cytosol, endoplasmic reticulum, free ribosomes, bound ribosomes. Notice that. Closer look. TM showing ER and ribosomes. Notice ribosomes here, and closer look, and large subunit, large and small subunits. Okay, thanks for listening. Nuclear again, nuclear lamino TM, the network lamino lines the inner surface of the nuclear envelope. Okay, thanks for listening.